Physiological effects of auxins. First, isolation. Auxins are synthesized in different areas. First, auxins were observed by Charles Darwin and Francis Darwin and isolated by Wendt. Here, isolation, it is by F.W. Wendt from Avena coleoptile or oats coleoptile tip. Next, here from the plant, these were isolated, and before the isolation of auxins from plants, the auxins were isolated from human urine. Here from oats, auxins were isolated, and before the isolation of auxins from oats, the auxins were also isolated from human urine. Next, occurrence. Here, where they are synthesized, auxins were synthesized in here root apices and shoot apices, shoot apex and root apex. So, in the apical portion of root and shoot system, here the auxins were synthesized. Auxins are synthesized and here these are transported. Here in the root apical, the shoot apical meristem, the root apices also here synthesized, and from the synthesis place, here the auxins are synthesized from the synthesis site to other places, and downwardly here these are translocated to upwardly. So from the synthesis place to here different locations, here vice versa, they are translocated. Or the synthesis is taking place in the shoot apical meristem or shoot apex and root apex. Next, types of auxins. Here the auxins are mainly of two types. Here natural auxins and artificial auxins. The auxins are of two types: natural and synthetic or artificial. Natural auxins are here indole acetic acid, indole butyric acid, indole acetic acid, indole butyric acid. These are synthesized in the plants. So natural auxins. Synthetic auxins are here, these are artificial auxins synthesized in the labs. For commercial purpose, they are synthesized. Here 2,4-D, 2,4,5-T, 2,4-dichlorophenoxyacetic acid, 2,4,5-trichlorophenoxyacetic acid, and naphthalene acetic acid. So these are the artificial auxins or synthetic auxins. They are synthesized in the plant body, and these are synthesized in the lab. 2,4-D, 2,4,5-T, NAA, 2,4-dichlorophenoxyacetic acid, 2,4,5-trichlorophenoxyacetic acid, naphthalene acetic acid. Physiological effects of auxins. Here, these are the physiological effects: abscission, apical dominance, xylem differentiation, cell division. So one by one we will discuss abscission. Abscission means here the falling of leaves or falling of fruits or shedding of leaves and fruits. So leaf drop, fruit drop, it is nothing but abscission. But in the normal concentration of auxins, here the abscission is prevented. The normal concentration of auxins. In young plants, here the normal concentration of auxins, it prevents the abscission. Auxins prevent the abscission in young plants or in the very young plants. Next, abscission is prevented in young plants if the auxin concentration is normal, but in the low concentration of auxins, the auxin concentration is low, that is promoting the abscission, falling of leaves, shedding of leaves, it is promoted. If the concentration of auxin is low, it is promoting the abscission in mature plants, mature or old plants, the abscission is promoted. So here two different actions. Abscission is prevented, abscission is promoted. But in young plants, the concentration of auxin is normal or in high concentration. So it prevents the abscission in young plants, but if the auxin concentration is low, that is promoting the abscission in mature or old plants. Next physiological effect is apical dominance. Here apical dominance means apical bud. Apical bud is inhibiting the growth of lateral buds or here axillary buds. If you see one in plant, apical bud is present in the apex of the shoot system or apex of the apices of the branches. It is apical bud. Here the auxins are synthesized, and in the axils of leaves, the axillary buds or lateral buds are also present. Lateral bud, axillary bud is present. So here auxins are synthesized in the apical bud, in the apex of the shoot system, and here it is not present, it is translocated to different parts. So here it is translocated to axils or axillary buds. So these axillary buds are receiving the here auxin. Auxin is synthesized. Only synthesis place is apical portion, but it is not present in here. It is translocating to axils or axillary buds. So axillary buds or lateral buds are receiving the auxins. When these are receiving the auxins, so these cannot develop into branches. So here apical bud is synthesizing, or in the apex of the shoot system, the auxins are synthesized. So it is translocated to axillary buds. So in the high concentration of these auxins, the lateral buds cannot develop. So when they receive high concentration of auxins, the axillary buds cannot develop into branches. So these are not developing into branches. No branches are developing, or very short branches are developed. Only it is showing the growth. Here apical bud is showing the growth, but lateral buds are not showing the growth. Why? Because they are receiving high concentration of auxins. High concentration of auxins, they cannot develop into branches. So it is dominating lateral buds. Apical bud is dominating the lateral buds. So it is called as apical dominance. The apical bud is present, auxins are synthesized, and these auxins are translocated to lateral buds. In the high concentration of auxins, here these cannot develop into branches. So only apical bud is showing the growth, no lateral growth. So lateral growth is inhibited by the apical bud. So it is called as apical dominance. So this apical, how can we remove the apical dominance? So if we remove the apical bud, here apical bud is not there. So if we cut the apex of the shoot system, here no apical bud. So no synthesis of auxins. Here no auxins are present in the, here no auxins are translocated to the axillary buds. So here in the less concentration of auxins, these are developing into branches. It is developing into branches. So when apical bud is present, that is synthesizing the auxins and translocating the auxins to lateral buds. In the high concentration of auxins, they cannot develop. But if we remove the apical bud, decapitation or apical portion is removed. So if we remove the apical portion, no synthesis of auxins. When the apical bud is removed, here no synthesis of auxins, here no translocation of auxins, here in the less concentration or in the absence of auxins, the lateral buds can develop into branches. So here by removing the apical bud, we can here remove the apical dominance. It is also here affected by the auxins. Next is xylem differentiation. Xylem differentiation means the cells are dividing in meristematic tissues. The meristematic portion in the stem, here cambium, the stem, if we take a cross section, we can observe like this in dicot plants, dicot stem. So vascular bundles. The vascular bundles in the vascular tissues, here xylem and phloem are present. Xylem and phloem together called as vascular tissues. Here how the xylem is differentiated, how the xylem is formed. So the meristematic tissues are cambium. The cambium is dividing and forming the new cells. The new cells are here undergoing some change. So after undergoing some change or differentiation, the xylem is formed. So newly produced cells are living cells, but after some time they become as dead, means the protoplasm loss. After the removal of protoplasm, they become dead cells. So in the xylem differentiation or in the xylem formation, here the auxins are involving. Next is cell division. So in addition to cytokinins, here auxins also involved in the cell division. Here cell division or kinesis. So these are the physiological effects of auxins: abscission, falling of leaves and fruits, fruit drop or leaf drop, apical dominance, apical body showing the dominance on the lateral buds, xylem differentiation, so the formation of xylem. Next, cell division, kinesis, cytokinesis. So these are involving. Next, applications, how they are useful to human being or farmer or in the gardening or in the horticulture. Here rooting, flowering, parthenocarpy, weedicides. Rooting, applications of auxins. First of all, rooting. Here rooting means formation of roots. Here stem cuttings. Here if we cut the stem or internodal portions, here all these are stem cuts. So stem cuts are when they are dipped in the auxin. So this terminal pore, the apical, the total stem, it is cut into small pieces. The small cut portions are stem cuttings. The stem cuttings when they are dipped in the auxins, so from the terminal part of the stem, here roots are formed very fast. So auxins are promoting the rooting, root formation. So like indole butyric acid or auxins generally promote the root formation from the stem cuttings. So it is useful in the formation of roots. So we can produce number of plants within a short period. So here the root formation becomes very fast in the stem cuttings. Next, flowering. Flowering is also promoted or sometimes inhibited also. Here flowering is promoted in pineapple, in the low concentration of auxin. If the auxin concentration is low, that promotes the flowering. So in pineapple, flowering is promoted. Next, in lettuce plant. In lettuce plant, flowering is are stopped or inhibited. In pineapple, the fruits are important. Here, in the low concentration of auxins, here promote the pineapple flowering. So the flowers are here after fertilization they develop into fruits. In lettuce, flowering is inhibited. It is also useful. So if the flower formation is inhibited, how it is useful? In the lettuce plant, only leaves are edible. Here, leaves are edible. So in lettuce plant, commercially, here the flowers are not important. Here leaves are edible. So if more leaves are produced, that is beneficial to the farmer. So here low concentration of auxin, it promotes the flowering in pineapple. High concentration of auxins, or here the auxins are sprayed. High concentration of auxins are sprayed on the lettuce plant. Flowering is inhibited or stopped. So here more leaves are formed. So leaves are only edible. So in it is showing different effects in pineapple and lettuce plants. Next, parthenocarpy. A parthenocarpy means without fertilization. Without fertilization, the ovary develops into fruit. It is called as parthenocarpy. In tomatoes, parthenocarpic fruits are formed. So when the flowers are sprayed with auxins. So on flowers or on the crop, if the auxins are sprayed, so the ovaries develop into fruits without fertilization. So without fertilization, if the ovary develops into fruit, no seeds are present. So seedless fruits. If the seedless fruits are formed, so that is commercially it is beneficial. Sometimes grapes also, seedless grapes. So these are seedless. After pollination, fertilization, if the ovary develops into fruit, that contains seeds. But here without fertilization, the ovary develops into fruit in these crops, tomatoes and grapes. So this parthenocarpy is promoted by auxins. So in these plants, here seedless fruits are formed if they are treated with auxins. Next, weedicides. 2,4-D, 2,4,5-T, these are used as weedicides, weed killers. So these kill the dicot plants. So these are used in the monocot crops. These are also auxins, kill the dicot plants. So these are used in the monocot crop. It is about physiological effects of auxins.